class because of Labor Day, so I'll be seeing you again on Wednesday, but not Monday of next week. And today we're going to continue dealing with how the cranial structures are used to move fluids, and we pretty much dispatched how ventilation works. We talked about how various spaces have changes in volume. Those changes in volume in turn cause changes in pressure. And then depending on the hydrostatic pressure gradient and whether certain structures are open or closed, you get flow of the water. But what you still have to wrap up for ventilation is what are the muscles doing in order to attain that end. And here's a little schematic diagram of a bony fish. What's this collective structure? Neurocranium. This is what? This. Hyoid arch. This. Okay. And this. This. Okay. And this. Good job. And can you all sketch this by now? Okay, because that's a good way to remember these spatial relationships. Okay. And remember that there are two ways in which the water is moved over the gills. One is pushing it by using a greater positive pressure in the oral cavity compared to the opercular cavity, and that's referred to as the forced pump. And remember, in the forced pump, you have a decrease in the buccal volume that increases the pressure and forces the water across the gills. This is the Abductor Mandibula. And what does it do during the forced pump phase of ventilation that's important to ensure that the water flows in the proper direction?
Closes the mouth.
Keeps the mouth closed. So it closes that most anterior opening. So then water can only go through an opening. You don't have an opening at the front of that chamber during the forced pump mechanism. Okay? This muscle that goes from the tips of the lower jaws to the medial portion of the hyoid arch is the genial hyoid. It's active during the forced pump, as is the sternohyoideus, which is another muscle with fibers that go along the length of the animal, and it goes from the hyoid arch back to the pectoral girdle. And to a close approximation, those two muscles are on at the same time. What do you think they do? So there's like this muscle attached here, this muscle attached here. They're both attached to the anterior, sort of mid-ventral region of the hyoid arch. No.
If they were pulling the hyoid arch and the pelvic girdle forwards, then wouldn't it be reducing the volume in the opercular cavity?
They are involved in reduction of volume. For all practical purposes, the pectoral girdle is just a big, robust anchoring region that's not moving. And it's such a hefty region, it's these smaller structures that are moving.
Okay.
What's the shortest distance between 2 points? Straight line. Let's consider 2 points. A point here and a point here. Is that a straight line? Okay.
Does it move the hyoid arch up, like push it back?
Exactly. Pretty tricky. So when both of those muscles are active at the same time, they're going to shorten, okay? And this is elevated. And of course, the hyoid arch is sort of embedded in some soft tissues. So in addition to just the hyoid arch itself being elevated, think of that as elevating the floor of the buccal cavity. Okay? Which is going to decrease volume and contribute to increasing pressure. All right? And then a muscle not shown in this diagram, because this is a lateral view, is the Adductor Arcus Palatine. So that would be on the medial side of the suspensorium. So again, if you think of these as the suspensoria, and this is the brain case, and you've got a muscle here, what's that going to do? Precisely. It's going to compress. It's going to adduct. It's going to move those two bony plates closer together. It's going to push on the water and reduce the volume. Okay? I'm not going through every single muscle and how it's used. I'm sort of focusing on some of the biggies. So realize this is a little bit simplified because fish heads are pretty doggone complicated. Okay? Now, the other phase of the ventilatory cycle involves pulling the water across the gills. You have negative pressure in both of the chambers, but there is a larger magnitude negative pressure in the opercular cavity, which is behind the gills. So even though you've got a negative pressure here and a negative pressure here, this smaller magnitude negative pressure is greater than this. So you have a hydrostatic pressure gradient that is still in the same direction, but it's generated by a different mechanism, namely increasing volumes and decreasing pressures. Okay? And the biggie for doing that is the levator operculi. So that's shown here. That's going to sort of cause this pivoting movement of the opercular series of bones, which is going to expand the opercular cavity volume, and hence generate a very large negative pressure. Interestingly enough, in the suction pump phase, once again, the sternohyoideus muscle is used, but it is not used at the same time as the geniohyoideus. So if that muscle is active, what's that going to do?
Depress the hyoid.
Precisely, okay? Because again, here's the pivotal point for the hyoid. It's going to depress it because of that initial starting position where the distal portion of the hyoid is anterior to this pivotal point. So that's going to go down and backward and hence again expand the bottom of the space enclosing the opercular cavity. And then again, a muscle that's not readily illustrated in the lateral view is the Abductor Arcus Palatine. So once again, if these are the suspensoria, we've got a muscle that goes like this. And abduction means move away from the middle of. So they move out like this, which expands the volume in the buccal cavity and generates negative pressure. But that expansion and subsequent subsequent negative pressure is not as large as the negative pressure in the opercular cavity. Okay? So I think it's really interesting that you've got sort of a context that's important for determining what the sternocleidomatus muscle does. If it's active with the genial hyoid, it actually has the reverse function as when it's active just all by itself and depressing the hyoid arch. Pretty tricky how it gets used in those two different ways. Okay? And as I mentioned, a whole lot of fish suck water in when they feed. There are some other ways of feeding, but that's a very common way of feeding. So actually, now that you've got a good handle on how water gets sucked in during ventilation, basically what happens during feeding is what happens during the suction pump, but happens really, really fast, and it's very powerful, very impressive indeed. So we're going to spend the remainder of today dealing with feeding of fishes. And at the level of detail that we're going to go over in lecture and is on your handouts, once again, you want to keep track of what moves, and if we explain what muscles move structures, know those. And there's a distinction made between suction feeding and ram feeding. And this is going to be one of many examples that we're actually going to have of, you often see, let's put things in box A and box B. But then there's kind of stuff in between box A and box B, okay? So I think one thing that's really useful to do in these circumstances is recognize that there may be some terminology for two extremes, but those extremes may actually be along a continuum of variation, okay? So don't let that fool you. But historically in the literature, a lot of this has been dealt with as an either-or issue rather than how much of these two types of feeding. Okay? And again, at the level we go over in things on your handouts and here in lecture, yet another recurrent theme in here is enhancing your ability to infer function from structure. So a lot of what we're dealing with in here is how is structure related to function. And certainly when it comes to bones and muscles, ultimately you need to do experiments to pin down how things work. But a lot of times you can make very reasonable inferences based on simply how things are built or how they will work, and how good they are at doing one thing as opposed to another. Okay? So I mentioned a lot of fishes use suction feeding. How important it is does depend on the taxonomic group. And I'm going to be painting with some pretty broad brush strokes here, so take that into account, because as you are painfully aware by now, there's certainly a lot of species of fishes. So allowing for sort of these gross generalizations, I think it is fair to say, if you compare the cartilaginous fishes to the bony fishes, the use of suction feeding is much less important. That doesn't mean it's absent, but when you actually look at that capacity in bony fishes compared to chondrichthyes, it's pretty darn impressive. And there are a fair number of cartilaginous fishes where suction's not important at all, okay? In some of those chondrichthyes where it is important, it used to just be that folks thought it was completely unimportant for chondrichthyes. But then, you start looking at the diversity of different sharks and their relatives, and in fact, some examples have been found where suction feeding occurs. So is this an example of suction feeding? So there you've got a left lateral view, and this was in a transparent tank, so you've got a mirror view as well. Is that suction feeding or not in this little bottom dwelling shark? Who says it's suction feeding? Raise your hands. Who says it's not suction feeding? Okay, then the follow-up is, why do you deem that suction feeding?
Because he's creating a difference in pressure by changing the volume of his, what that's called.
Okay, well, what if the fish is moving forward and it has its mouth open? Won't that push water in the mouth and expand that volume? So is that by itself sufficient information? Okay, that's a correlate of suction feeding.
Like the background, he doesn't move against the background. Like he's staying in the, or if he does move, he moves very little.
He moves very little, but what's even more important than him moving little
It's the,
is the food item does move, okay? So he generates enough suction to actually pull the food item in, okay? That's the key criterion. That's the acid test for whether or not it's suction feeding. Because where it gets tricky is sometimes the fish will keep moving and it will still suck the prey in. That's where it gets tricky. Now the other extreme in ram feeding, you got a food item here, you got a fish here, no movement of the food item at all. Basically the fish swims through the prey item and then closes its mouth. Tunas are a great example. Very fast moving fishes, they just gobble other fishes down as they swim rapidly. Okay? And then here are some stills from high-speed video of Polypteriformes. Polypteriformes is quite interesting because among extant bony fishes, it has many traits that are believed to resemble the ancestral condition for a lot of bony fishes. So if you look at where this little, I think that's a mealworm was, relative to those spots in the background, in fact, the mealworm gets drawn into the mouth. But if you actually look at what bones move, even though the buccal volume increases as the mouth is opened and then sucks the mealworm in, you don't see much in the way of movement of either the maxillary or the premaxillary bones in Polypteriformes. Okay? And then another fish that is not a teleost fish, that doesn't have a homocircle tail, are the bowfins in the genus Emia, the order Emiiformes. They do suction feeding as well. And in their case, the maxilla has a good amount of mobility because this would be nearly horizontal at rest. But the premaxilla is not mobile, okay? So one of the things we see in the more derived condition for bony fish heads is a bunch of stuff moves. Unlike our skulls, there's just all kinds of stuff that moves in bony fish heads. Okay? And you have a lot of the key steps for suction feeding, sort of the more generalized type of suction feeding in bony fishes on handout 5, page 3. And if you look at your cladogram for where Acanthophora rigii are. The vast majority of Acanthophora rigian fishes are the purplemorph fishes. Remember, the purplemorph fishes are easy to spot because the pectoral and pelvic girdles are fused. So instead of a big distance between those two sets of paired fins, as you'd see in something like a salmon or a sturgeon or fishes like that, they're practically right next to each other. Okay? And suction feeding has been studied very extensively within the Acanthophora rigii and within the purplemorphs in particular, which are a subset of this group. And in a whole bunch of these, you see a highly mobile premaxillary bone, unlike in the bowfin fish. So instead of the jaws just sort of opening and closed, the jaws can actually be protruded. But precisely how much they're protruded depends upon the species. Okay? So this is something that's highly variable, but in some cases can actually be quite extreme. So a whole bunch of stuff moves when fishes perform suction feeding. Okay? And I showed you this one high-speed video clip previously. Let's just look at it again. Trying to keep track of some of the things that move. Look at the dorsal surface of the head relative to the rest of the fish. Or the goldfish. And what's going on with the neurocranium?
It's elevated.
Okay. So when fishes open their mouths, at least these fishes, it's not just a matter of here's the skull and then you open the lower jaw. That's what we do when we're flapping our mouths, although no one can see it anymore because we're all wearing masks, okay? But when fishes open their mouths, they have a considerable amount of cranial elevation in addition to depressing the lower jaw relative to the brain case. But then also look, here's the premaxilla, okay? And here's the premaxilla before mouth opening. So not only do you have the mouth opening, but you have these most anterior bones, also known as the oral jaws, moving forward relative to the brain case. And this is a very widespread condition in Acanthophorigian fishes. Now, a lot of you have probably seen bluegill and perhaps even gone fishing for bluegill, in part because they're readily available. They've almost been like the lab rat for studying locomotion and feeding function. They're little piglets in the lab, so they feed very, very readily. Okay? Okay, just to orient you, this is a left lateral view of a bluegill in the transparent tank. There's a little piece of earthworm. And then once again, we have a mirror view so we can see the ventral surface of the bluegill. And these are pretty much lined up relative to each other. And as I mentioned, suction feeding happens very fast. So realize this is slowed down tremendously because actually the last digit in these numbers is 1/1000th of a second. Okay? So there's the food item. There's the fish. We've got to keep track of who's moving and who's not. Did the earthworm get moved? Yes. So the earthworm got sucked in. Now, the bluegill, almost many suction feeding fishes almost come to a dead stop. It's not always completely coincident with the time of maximal suction, okay? But one thing you'll notice is the bluegill is sort of slamming on the brakes, if you will, by moving those pectoral fins forward. And what do we see here that was not visible just a little bit earlier?
The abduction.
Indeed, there's abduction that is present in the lateral view. In the ventral view, what do we see?
The oil bones coming out.
Yes, the oral jaws are being protruded. So notice how much they're actually sticking out relative to that rest position. Okay, let's move back. So notice here's the shape of the fish head. There are the eyeballs sticking out. There you can see very clearly the jaws protruding. What about the width of the fish in this region and this region? Increase or decrease? Increases, okay? So besides the ventral portions of these chambers being lowered, you also have this lateral expansion of structures that generates the suction, okay? So as indicated on your handout, you get mouth opening with protrusion of the jaw. And that initial mouth opening is happening when the opercular valve formed by the branchiosicle is closed. And as we also mentioned, there's some cranial elevation that is involved in opening the mouth, as well as depressing the mandible. And one of the things that's pretty neat is there actually is, when we open our mouths, it's a simple thing because we have a jaw joint here, roughly, and we have a muscle that attaches behind a pivotal point. So that muscle shortens and then it pivots our lower jaw down, okay? If bony fish is opening their mouths, it's a complicated thing. There's not anything that is strictly similar to that. You actually have these indirect mechanisms of opening the jaw. And these very powerful muscles that move the opercular series of bones, they pivot them so the ventral portion moves posteriorly, and then because of that ligamentous connection, that actually opens the lower jaws. So oddly enough, two of the major mouth opening muscles during suction feeding are the levator operculi, and then simultaneous activity of the hypaxial muscles and the sternocleidomastoid muscles, which are also involved in expanding the ventral surface of the buccal cavity. Okay? And then a little bit later, you see this additional expansion via hyoid depression and expansion of the suspensorium. Now, you can't see that too clearly in that particular video. And then as the then you basically just have things reverse. When the mouth closes, unlike mouth opening, as indicated on your handout, 5 on page 3, that's done directly by the Abductor Mandibula muscles rather than an indirect mechanism. Okay? And then, as the mouth closes, you basically have a bunch of events happen, as elaborated on page 3 of handout 5, that compress these various spaces and eject the water out the gills. So They suck food plus water in, they don't swallow the water, okay? The prey item is retained and the water is expelled via the opercular valves, okay? I guess one shouldn't have favorites. I mean, as a parent, you're told, you shouldn't have a favorite kid. Perhaps as a biologist, you shouldn't have a favorite critter. I got a weak spot for this one. This is a sling-jawed wrasse. These are found in the Pacific. These are reef fishes. Wrasses are extraordinarily diverse for the shapes of the head, what they eat, and exactly how they eat. And one of the reasons this is my favorite fish is because talk about jaw protrusion. Now we've really got some jaw protrusion going on here, okay? So these are some stills from some high-speed film. Yes, this was back before the days of digital photography and people actually used film. Notice this is the start of feeding, and 30 milliseconds later, the jaws are fully protruded, okay? So think of that, that's 30 milliseconds, that's roughly 1/30 of a second. Remember, if an eye blink is 1/5 of a second, that's 1/6 of an eye blink. That's pretty fast, right? And not only is it fast, it's just an incredibly large amount of jaw protrusion. And it's not completely clear why some species have highly protrusible jaws and why others don't, although there have been some reasonable suggestions for why that may occur. It may be that if you form this long tube-like structure, it sort of enhances this efficiency of this regime of generating flow via negative pressure. So it might be hydrodynamically more efficient. And then it also may be effective in sort of catching the prey and actually blocking the escape of the prey. The prey quickly is enveloped on all but one side by this expanding tube. Perhaps that's what's going on. But this is still an area of active inquiry. So again, this is slowed down tremendously because I want you to remember that it only takes about 1/30 of a second for the jaws of this fish to protrude maximally. Okay? I mean, how can you not like that, right? it's just so bizarre looking. Okay? So, this is not like our heads, okay? And I cannot emphasize too much this profound difference between the heads of bony fishes and the heads of mammals, which are pretty boring. You know, we basically have a rigid brain case and a simple jaw that flops beneath it. But a whole bunch of bones move in fishes, and all of those movements are put to good use for both feeding and for ventilation. Okay? Well, you're well aware that fish have teeth. You've seen this in lab, and probably just, from your everyday knowledge, you realize that fish have teeth. Fish can have a lot of teeth, and a lot of teeth on a lot of different bones. Now, what probably isn't too surprising to you is that those oral jaws commonly, not always, but commonly have teeth. And what are those bones involved in the oral jaws? Kind of three biggies. What's the upper jaw bones?
The maxilla and the premaxilla.
The premaxilla and the maxilla, and then the lower jaw? And the dentary within that collective structure that makes up the mandible. okay? So, that's not that radically different from when you kind of look at our mouths and what's going on. But many fishes actually have teeth on the gill arches, so-called pharyngeal teeth. So besides teeth on the oral jaws, sometimes you'll actually see the gill arches being referred to as the pharyngeal jaws. And how big those are, how they're shaped, all that has consequences for what those structures may be good for, okay? So this is something that pops up in teleost fishes. Remember, teleost fishes are those fishes with almost circle tails. And it might even be yet another synapomorphy for that very large group of bony fishes known as the teleost fishes. Okay? So again, let's think about how things are built and some of the implications for how things work. Structure, function, relationships. Big recurrent themes. Okay? Not all teeth and fishes are created equal. Here are the teeth of a great white shark. I think most of you are well aware that they're very adept at taking chunks out of all sorts of things, whether it's from a fishing boat that's trying to catch them, as in the movie Jaws, or perhaps from the prey that they're going to consume, and so on. Look at the teeth of the piranha, just to the right, okay? So actually, in both of these cases, we have these rather triangular blade-like teeth, and when they close, they tend to sort of interdigitate, okay? And that's actually very good for cutting pieces of stuff, okay? And both of these species are very good at taking bites out of their prey. Now, in some cases, and you've seen gar in the lab, those teeth are not remotely blade-like. They're sort of needle-like or more peg-like. Now, they're still good for doing some stuff, even though they're not good for cutting. Namely, they're good at puncturing surfaces and then penetrating surfaces, so they're pretty good for hanging on to things, but not well suited for actually taking bites out of things. Okay? And then you have some teeth, unlike those first two types, that far from having a sharp tip of a blade or a sharp needle-like point, just big old fat, robust things with flat ends. Presumably, that reminds you of the molars in your own mouth. And those structures are good for crushing things. So many bottom-dwelling fishes that eat a variety of things that are hard-bodied, like crustaceans and so on, that's kind of tough to deal with. But if you just smash that exoskeleton, that can be very helpful indeed. Okay? Parrotfish are pretty cool. I think probably a lot of you realize that these are fishes that are very common in the Pacific and associated with coral reefs. And just look at these massive pharyngeal teeth that you find within the mouth of parrotfish. Does anyone know what parrotfish eat? At least many of them?
Oh, don't they like the coral?
They do, okay? So coral sort of has two parts. There's a living part of coral, but of course coral basically builds up rock around it. That's what limestone is. from reefs, right? Well, that's a pretty crazy thing to be eating. Just think of that. They're basically crushing rocks to get to the living part of coral.
You go to the beach, you might just be laying in parrotfish poop. Parrotfish have great big teeth. They chomp and eat coral, grind it up to get to the algae living inside. They are vegetarians. They don't digest the hard coral, only the algae. So when parrotfish poop, it is trails of sand.
You see that cloud of sand as the fish are swimming along. So I thought that's kind of a cute video. The whole notion that you go to a beach in these areas and you're actually laying out on parrotfish poop. Now, obviously, other things break up the reefs and cause the sand and so on. But the amount of material that they have to process is really amazing. And it shouldn't be too surprising then that you actually see a very specialized structure associated with that specialized function of basically crushing rocks, okay? So that's certainly one of the extremes, okay? You can also get some hints about how fish are feeding from the size and location of the mouth, and also where the fish is actually feeding. So if you think of some of the things that can vary in the feeding ecology of fishes, of course, one thing is what's being consumed, okay? There are many different things that are being consumed. But then another thing is what tactics are used in order to exploit that resource. And part of that equation is where the stuff is actually located, okay? So if you think of fish moving around, if we transformed this classroom into an aquarium and filled it with water, then fish could get to any place in here, right? Whereas right now, because of gravity and all that stuff, we are bound to the floor, okay? So one thing that fishes have to deal with is dealing with the profound consequences of resources and how they're distributed in 3 dimensions, okay? And that gets pretty complex. And not all fishes use all of the volume around them equally, okay? So for example, there are some species that tend to feed near the surface. Other species may ambush the prey from below, okay? And in these cases, you'll see that the mouth has a so-called superior orientation. So instead of, you've got the long axis of the fish here, and instead of the mouth just being here, it's sort of oriented here. And the mouth opening is pointed up, okay? Here's an example. This is a stargazer, and notice how dorsally that mouth opening is oriented. Here's another example. Boy, that's a weird looking fish.
Writing's not that easy, but Grammarly can help. This sentence is grammatically correct.
Okay. Again, this is super slow motion. These frogfishes and their relatives, the antennaria fishes, are sort of chubby things very often, hang out on the bottom, and actually walk on the bottom. So if they're on the bottom, it stands to reason a lot of stuff is actually going to be above them. So it helps to have that dorsally oriented mouth. Okay. There are some other fishes where the mouth is basically right at the end of the line, okay, the most anterior location. And very often, these fishes are actively swimming around and catching very mobile prey. Oftentimes, those mobile prey are other species of fishes. Tunas are, and the relatives are just really impressive.
At some times of the year, seasonal changes make the currents especially rich in nutrients, and then the ocean around the seamount becomes a virtual soup of plankton. At such times, hunters gather in astonishing numbers. Bonito, smaller relatives of the tuna. They're searching for still smaller plankton feeders that have been attracted by the bloom. So are these jacks. Their prey is nearby. A school of anchoveta has strayed up near the surface, even though it's broad daylight and hunters are on the prowl. These small fish can already feel the vibrations of the approaching predators. Swimming at speed, they formed into a ball, and now they must wait for whatever comes.
Pretty amazing collective defensive. Let's see how effective it is.
At first, the sheer scale of the great ball seems to daunt the predators. But now the bonito arrive and launch the first attack.
Notice how rapidly the predatory fish are swimming. So they're basically using that speed to gobble up the fish.
The young yellowfin tuna move in. The speed of this attack is so great that gradually groups of anchoveta are splintered from the main fish ball.
The incredible speed of the attack. So that's a pretty classic example of land. Remember how many fish there were? Pretty amazing. Even the leftovers, they're gone. Okay? Great example of ram feeding. Very rapid swimming. Suction is basically insignificant. It's just that high-speed swimming of the fish opens its mouth and puts the fish. Well, sort of on a less spectacular scale, perhaps. There are some bottom-feeding fishes. We've seen some examples of these in the lab, including various suckers and sturgeon, where instead of the mouth being terminal, it's oriented ventrally. So it makes a lot of sense for the fish being slightly above the bottom. It can maintain a horizontal orientation and still exploit resources that are beneath it. And you can have a variety of different shapes of fish heads, different advantages and disadvantages to each Some fish are remarkably adept at exploiting very small prey items. One of my favorite examples of that would be the long-nosed butterfly fish shown here. So it's got that incredibly long tubular mouth that can get into the nooks and crannies and coral reefs and use sort of plankton size prey as a result of that. Okay. Some additional structures that are correlated with function are the Gill Rakers. Depending on the species of the fish, and sometimes even the population of fish within a species, the numbers of these structures may vary, how long they are may vary, how thick they are may vary, and how closely spaced they are may vary. And that variation, in fact, is correlated to what's being consumed, the type of prey, how big it is it hard or squishy? Okay? And actually, these illustrations are from remarkably different populations within the same species, where here you see a piscivorous population. Here you see an example of something that is still a predator, but on relatively small prey. And then at another extreme, here are the Gill Rakers. So the rakers are the structures to the left of the gill arch. These are just the gill filaments off to the right. And obviously, this can form a fine sieve. And a good example of a fish that feeds on plankton is the very bizarre paddlefish.
Hi, my name is Jim Adams. I'm a biologist here at Sunrise Aquarium, and I take care of this paddlefish dive to the bottom. It doesn't have that level nose.
And here you can see one swimming around its tank, this huge mouth open, and it has very fine gill rakers that basically form a sieve. And so even though these can be pretty sizable fish, they're exploiting a very small prey size. And they're able to do so because of this anatomical specialization. Okay? Now, besides all this variation up in the head, it shouldn't be too surprising that different fishes are eating different things. Their digestive tracts, in fact, may differ. Again, if one paints with some very broad brush strokes, herbivorous species of fishes very commonly have a long gut and a highly coiled gut, as shown here. Okay? Whereas this can be quite short in carnivorous species, and then one thing that kind of blows me away, there are actually some fishes that don't have a stomach, okay? And that occurs in some planktivores, and some herbivores, and some fish that are actually parasitic. Okay? And here's just a little diagram that shows you some of the diversity you can find within a single evolutionary lineage of fishes. And this is sort of a classic example. The African cichlids became very diverse very fast. And of course, at the community level, potentially one problem you have with many species is if you have a whole bunch of species, are different species actually competing for the same resource? So as the species diversity increases, a priori, you would expect to see greater specialization in species, so they're not competing for the same resource. And without worrying about a whole bunch of the gory details, if you just look at the different shapes of the teeth, the head, the size and orientation of the jaws, be convinced that actually, in addition to having these differences in diet and feeding ecology, that's actually correlated with different structures, different structure in the trophic apparatus of these fishes. And presumably that's been that plasticity, that evolutionary plasticity of the trophic structures may have actually been a key thing that facilitated so many species evolving so rapidly. So clearly, a lot of species of fishes, they eat a lot of different things. We could basically have a whole course on it, but this is sort of the bird's eye view of some of the most general trends. Make sure you keep sort of reading handouts and so on ahead of time, as we're going to make a transition to how fish swim in the upcoming lectures. So do look for those handouts ahead.